Hello friends, today we'll discuss Enterobacteriaceae and E. coli. Enterobacteriaceae are gram-negative bacilli which resides in intestine. This family is grouped together and they possess some common characters. The common characters are they are enteric gram-negative bacilli, they are aerobes or facultative anaerobes, they ferment glucose with or without production of gas. They are non-fastidious, that means they grow on routine or ordinary culture media. They reduce nitrate to nitrites. They are catalase positive. The very important character of family Enterobacteriaceae is they are oxidase negative, gram negative bacilli. Please remember that. Most of them are motile and flagellated. The family Enterobacteriaceae has been classified way back in 1986 by Ewing, and as per the classification, the family is divided into 8 tribes. Tribe 1 Escherichia, Tribe 2 Edwardsiella, Tribe 3 is Salmonella, Tribe 4 Citrobacter, Type 5 Klebsiella, Type 6 is Proteus, Type 7 Yersinia, and Type 8 Irvinia. What I said is an important member of the genus, while tribe is pronounced like Escherichia. Now we'll discuss E. coli. Once again, they are a very important member of the family Enterobacteriaceae. It was first identified by Theodore Escherich in 1885, and they are most important pathogen among the family. They are also one of the most common aerobe found in gut of human and animals, and they are excreted in feces, and when such feces contaminate water, this organism remains viable for few days, and that is why they are considered as indicator for fecal contamination of drinking water. Now this genus Escherichia coli has got certain virulence factor. You can see over here in this figure, this is a figure of a gram-negative bacilli having certain components outside, which you can see a flagella, fimbria, then a capsule, cell wall, and inner bacterium. So outer side there are certain antigens, and these antigens are called as surface antigens. The first one is the somatic antigen, O antigen, that is on cell wall. The second one is flagellar antigen, that is present on flagella, also called as H antigen. And third one is capsule, K antigen. And the fourth one is the fimbrial antigen, that is the fimbria, an organ of adhesion. Now we can see there are several different types, that is which organisms can be classified based on its reaction with specific antisera. Different type of O antigens are something around 174, H antigens are 53, and K antigens are something around 100. So we can identify the serotype based on commonly O and H antigen. As for example, O157H7 is one of the virulent strain of E. coli, which is responsible for hemolytic uremic syndrome. So we discuss further about the somatic antigen. What is that? They are lipopolysaccharide antigen present on the cell wall. They are heat stable, and when we go for a serotyping, this O antigen is not specific for particular species, rather genus, but they can cross-react with other member of the species. And so this is a cross-reacting antigen. Remember, O antigen is one of the most important virulent factor for the bacteria, and it possesses endotoxic activity. So after lysis of the cell wall component O antigen would lead to toxemia. And when present, this O antigen also inhibits phagocytosis and also inhibit complement mediated. Next we discuss about the flagellar antigen, H antigen. H comes from Hodge, and the literal meaning of that is a Greek word. The meaning is film of breath, that when we exhale our breath on a glass, it forms a smooth layer over that, a film of breath. So if such type of colony is seen on the media, this is why they are called as H antigen, because the presence of flagella is responsible for such type of appearance. And in case of O, that is Ohne Hodge, that means absence of that O antigen. Heat labile, remember this H antigen is heat labile, so in presence of a heat, they would be destroyed and will not be causing any pathogenicity. Motility, this as it is a flagellar antigen, it is responsible for the bacterial. Third is capsular K antigen. Some of the species contains a capsule. The word K comes from the Kaspel, that is German word, which means capsule. So capsule is polysaccharide in nature, and when capsule is present, usually it will mask O antigen. So when you go for serotyping with O antigen, O antisera, such type of strain is non-agglutinable. Once again, presence of a capsule hinders bacteria more resistant, especially in phagocytosis. Next is fimbrial antigen. We all know that is fimbria pili, organ of adhesion. It is also a very important organ for attachment and colonization. The common type of fimbrial antigen, that is CFA, colonization factor antigen. Other is P fimbria. Now such fimbria has got an attachment towards the P blood group antigen. So these are common surface antigen that we discussed. Now the toxins are produced by E. coli, and they are enterotoxin. The diarrheogenic strain of E. coli produces enterotoxin, and these enterotoxins are of three different types. One is the labile toxin, that is heat labile. Another is stable toxin, heat stable, and third is virotoxin or virocytotoxin. So remember that over here, enterotoxin, which is stable in presence of heat, is stable, while other is heat labile, that is it is destroyed in presence of heat. Apart from that, they also produce hemolysin, the virulent strain of E. coli. We'll discuss about the function and detection of enterotoxin along with the pathogenicity. Now, clinical manifestation of E. coli. The most common, they are responsible for urinary tract infection, and the strain which is responsible is uropathogenic E. coli. Other important disease which is produced is diarrhea, and according to the type of the strain, there are 6 different strains of E. coli which are classified, like enteropathogenic E. coli, enterotoxigenic E. coli, enteroinvasive E. coli, enterohemorrhagic E. coli, enteroaggregative E. coli, and diffusely adherent E. coli. We'll discuss this when we discuss the diarrhea in detail. Apart from that, it also causes some other infection like abdomen. Very commonly it is causing peritonitis, pneumonia, especially ventilator associated pneumonia, meningitis, especially in neonate, and wound and soft tissue infection in diabetic patient. Apart from that, it can also cause prostatitis, osteomyelitis, and bacteremia. Now we'll talk about general laboratory diagnosis. The first one is the sample. The sample would be obtained depending upon the disease produced. So it can either be a urine in case of a UTI, stool in case of diarrhea, or pus or wound swab in case of skin and soft tissue infection. The next is smear examination. After collection of a sample, they are subjected to wet smear examination or gram staining, where you can observe gram-negative bacilli along with the presence of pus cell. Then cultivation. The sample is cultivated on blood agar and McConkey's agar, and it is isolated and further identified by biochemical reactions like IMBIC, that is indole, methylrate, Voges-Prosker, and citrate, along with PPA, urease, and TSI, and sugar fermentation test. Based on the result of these biochemical reactions, the organisms can be identified. And the last step would be to perform antimicrobial susceptibility testing. Thank you.